Hello, welcome to today's coffee hour. It doesn't look like anyone's here this week, but that's okay. I wanted to go over some issues with people, make sure everyone's on the same page. If people want to contribute, I'll show you how to do that. We didn't have a weekly meeting this week because of prior engagement. So this coming Monday we will have a meeting, but we did not have one this week. So I have a couple of announcements for Ujwal and Mayuk, or GSoC students. If you have any questions about the first evaluation for GSoC, please let me know. It's not that big of a deal, really. You've been keeping up with your work. You've been making pull requests and commits to the repo, and it looks good. We've been regularly updating ourselves in the meetings. So I think you guys will pass pretty easily. And it's usually not a big deal. We usually, try to, as long as people are participating and showing up, they usually pass. Sometimes you have to give them some advice. I think overall you guys are doing very well. So I'll be filling out those forms this coming week, and you'll be getting feedback on that. So that's GSoC, and we'll revisit that periodically. I wanted to present on a couple things today. I wanted to talk about our repo and our issues, as I usually do. Then I wanted to talk about Neuromatch Academy, which occupied a good portion of my week, and I'll show you why in a minute. And then talk about maybe some issues related to the website and maybe some other miscellaneous things. So let me present my screen. Alright, and this should be good for everyone in case someone decides to join. So the first thing I want to cover here is the DevoWorm GitHub repository. And so I see over the course of this last week, GitHub changed their interface. So it looks, I don't know if I like it yet. I'm going to let it wash over me. I kind of like the old one better, but you always say that when they change something because it takes time for it to grow on you. But this is the new interface, and so you can see that they've kind of rounded the edges here. They've made things, I don't know, there's a certain design element here, sort of rounding the edges. This icon used to be kind of square, and now the quote here that you can put into your repo, it's excellent virtual science, that's now embedded in the image. So, and then the contribution board the icons are slightly rounder, so that's all kind of cosmetic stuff, but they've changed a couple other things too in the functionality. So people are looking at this repo and they ask what happened, then that's what happened. They changed it. So let's see, I'm actually logged into the wrong account here, but that's okay. We're not going to do any updating. So let's see. First thing maybe we'll talk about is GSoC 2020. So as you can see, we have mostly Jupyter notebooks in terms of languages. That's a little. The languages here are a little misleading because if you put up notebooks, like I know there are a lot of Colab notebooks in here that have Python and other languages embedded in them. So now they're claiming that most of the repo is Jupyter notebooks, but that's probably not the case. So we've gotten some commits, very good. Thank you, Ujwal and Mayuk. Ujwal is working on OpenDevoCell integration, so he's made some contributions this week. Let's go to the README on that. So, OpenDevoCell, platform to connect all logistics of DevoWorm group at one place. And so we have a standalone app for OpenDevoCell, which is it's a machine learning tool or deep learning tool that you can upload microscopy images to and have them segmented, and then get a readout of various parameters that the program extracts. And so this was something we developed last year in GSoC, and we are going to update that and transform it a bit. The OpenDevoCell aspect of it was sort of like something we did in the last week of GSoC, so it's sort of ad hoc, but we're going to make it much better. We're going to integrate it with the DevoZ platform, which I think I've shown before to people. This is a collection of data. So the idea is we're going to have this pipeline where we go from the DevoZoo to the OpenDevoCell, and then to some analysis that we specify later. But the whole thing, so people can come into the project or the organization knowing maybe very little about machine learning, maybe very little about biology. They can take some data, do an analysis, and then we can go from there. But the whole idea is that it should be highly automated so that anyone can come in and do something, and then we can, once we have a contribution, we can work from there. So we'll be adding to this perpetually. We started off with a couple of deep learning tools last year, working in our group with these things, and we've also have a longer tradition of computer vision tools that we've used, and so we're just kind of bringing that all together. I should say that, yes, and so Ujwal has a nice unified model diagram of his project here, as we've seen in maybe previous meetings. Here's the splash page. So this is a splash page for OpenDevoCell. I've already mentioned to Ujwal that I want to update the color. So the way it looks now is that all these splashes are coming up, and they're various colors, and we wanted to change this so that it's like a more uniform color scheme, so it doesn't pop up in your face, in radically different colors, and I found that a bit distracting. But we'll talk about the color template here in a minute. So this is Ujwal's OpenDevoCell, and he's made some contributions this week. Let me talk about I think I have the templates in here somewhere. Here it is. So if you go under definitions and concepts in DevoWorm in GSoC 2020, you'll find, of course, I think we've talked about these definitions before. Let's go into the color palette. So I've uploaded this color palette. There's a README here, and then there's this color scheme image. I don't know if I don't think I wrote down the actual thing that I wanted yet. I didn't finish. Oh yeah, I did. Okay, so this is a markdown doc on the color palette. So in my various endeavors that I've worked with, we have some color schemes that I've worked with and I like. And I learned this. The Elizabeth Warren for President campaign had a color template scheme that they shared with people for their campaign. It was like, they define different colors and their various shades, and those are the things that they used on all of the materials that they generated. And so that's something that you can use here. In the same principle applies. So I've created a bunch of colors that I've liked over the years in different outputs that I've had, and then I've defined them here, and below I've defined the RGB value or RBG values here. Red, blue, and green. So you have like the different colors, and you have this value, and you can convert these to hex. I could probably add another table of hex values for for Ujwal's benefit, but you can convert these online. So this is the idea that you have all these different color scheme. So OREL is the Orthogonal Research Lab. So you have light blue, dark blue, and then you have a standard white and a gray, which are, kind of neutral. You use them for, linking together the white and dark blues. So you might use it for the edges of the website or whatever. Synthetic Daisies is a blog that I've run. So you have different colors there: salmon, dark yellow, dark gray, and green. So you have all of these different shades of, these are the sort of the visual elements in the blog. So if you go to the blog, you'll see that it's yellow. There's some salmon elements, and then there's also this dark gray and green for background. And it just, these are the colors that I would like to see maybe in our background. For DevoWorm specifically, we have this purple, which is a nice color. And then OpenWorm has a green that's ubiquitous in the OpenWorm logo and then in other places. And then DevoZoo has its own blue-green. So I will work on these colors, and if people have other suggestions for good colors that, might look good in this scheme, because I like this color scheme. The reason I added all these different colors from different sources is I like all of these colors. They have a nice visual appeal, I think. And so, if we can just standardize it to one color or another, maybe two colors, we can make it look a lot less contrast there. So, definitions and concepts. Okay. So the color palette, we have some more of these definitions. So the meta features, we have this kind of, and I've not been working on this as much as I would have liked, but this kind of describes the meta features that we're looking for. As we go through this, I'm going to add this. I've added a couple references here, but I just bring this to people's attention because if you want to contribute to what's going on here, you can issue a pull request. So polysemi, it's still kind of I've added a set of lecture notes on this at the Orthogonal Lab, and that should be something that I can probably go through again and write down a little bit more here. But if you find something interesting in that set of lecture slides, or if you find something interesting here you want to expand upon, you can contribute. Okay, so now let's go over to what Mayok's been doing, which is the pre-trained models. Actually, he's been doing that and the worm movement. So this is the README for pre-trained models. So we have a lot of deep segmentation that's been going on here. The input image, prediction of this image, and the original mask. So what he's doing is creating a mask from this input image. It's kind of defining these yellow spots. So these yellow spots are the things you want to segment. You don't want to segment the yellow in the background. You want to segment the yellow in this blue cell. And so this is what's happened here. There's a mask where it's just kind of approximated either by hand or by dropping the contrast values and such. And then you get this image. And so this mask, you get, it looks like this. And then but you want to build a predictive model. So you want to use this to sort of train a predictive model that will pull these out. And so he's actually been able to do this pretty decently in this example. So he's been doing this data augmentation, which is where you stretch the data. So you take an image and you stretch it or you skew it in some way, and it gives the algorithm alternate views or alternate types of input, but similar to that image. So you can almost think of it as something adversarial, where you have an image, the idealized image, and then you have something in the wild that is maybe obscured by noise or has like a skew to it at a 45-degree angle. And so it'll learn that that's the same thing as your original image, and so it will pick up more variation. And so he's been doing this in, I think, PyTorch with the help of Lb Imitations. So what are Lb Imitations? I'm not sure. Take a look. This is a fast image augmentation library, easy to use wrapper around other libraries. Oh, so this is their team, I guess. It's not a term that I don't think I have to be familiar with here, but this is Lb Imitation is a GitHub repository. Lb Imitations, fast image augmentation library. Okay, so that's what that is. So he's using a standard library here, and so that's good. And the architecture is built on this ResNet 18 backbone. So he's sort of doing a variation on ResNet 18, and he's using this worm image dataset. So this is something we haven't used in our group. This is worm image, which is the database of anatomy, and it's actually has a lot of different types of data in it. It has from the embryo to the dower, which is a developmental form of C. elegans. It's a developmental, what they call a polyphenism. So a polyphenism is where you have different responses to the environment, and dower is something that is it's a response to like environmental scarcity. All right, let's see. So we have all those different types of images in worm image data set. And so he's using a variety of images here. We have all these larval stages in the adult, and we have different parts of the body. We have genotypes. So we have a very wide variety of things we can draw from here. We're not just interested in development, I guess. We would like to take this even into the adult. Would be in terms of developing the algorithm. I know main interest is development, but the idea is that we want to get as much training data as possible, and to build the pre-trained model for sort of we could, maybe it's C. elegans, maybe it's like just a general development. But to go in that direction, we need to find these meta features, which are things that are sort of unique to, maybe like microscopy data for worms or for developmental processes, things that you wouldn't pick up just by doing a quick analysis of shape or of like. Well, I guess it would be. This would be related to sort of the more abstract concept of shape, because if you're just trying to pick up features like, cells in pick cells out of a background, or pick organelles out of a cell, and people have been starting to do that. But if you want to find out more about like shape, and I think we talked about this with some of the morphogenesis research that's been done. If you want to know more about shape as an abstract concept and how it changes, then this is sort of the approach maybe we want to take. So in the spirit of that, we're doing the worm movement tracking metadata extraction. So this is where Mayuk is going to the OpenWorm movement database and picking up, these are adult worms that are moving on a agar gel, and they're doing their thing. And this is something that something called the Terpsy tracker that the movement group in OpenWorm have been working on. So he kind of took their code and he's building a lightweight version of that. And then, he's trying it on worm tracking. Obviously, that's what it's built for. But there are other types of movement we can get. There are movements in the embryo, for example, that you can analyze. And what better way to do it than to use something that we already know tracks worm movement. So it's actually pretty specialized for that type of adult movement. Maybe we can use it for the embryo as well. And of course, even if that doesn't work so well, and it probably should, but I mean, if you wanted to use it as a lightweight Terpsy tracker version, then that's also possible. So, and then this deep learning on cell position metadata, which is this is something we're revisiting the EPIC data set, which is a data set of the embryo. It's like where they track the cells, they track the cell position, they track the spatial aspects of embryogenesis, and he's revisiting this data set. We use this in the group. He's revisiting this data set so that we can extract, patterns within the data over time. And so it's just kind of a different way of looking at it. He's applied some principal component analysis, and there's some other goodies there. And so we're going along pretty well on this, I think. What do we move on from GSoC to our group repository? So why don't we go to the group repository and group meetings? And we got a lot of followers this week, and I'll tell you why in a little bit. We're up to 26 followers, which doesn't seem like a lot, but we had I don't know how many we had last week, but it was not that many. So I'll tell you why that probably is in a minute. But let's go on to the group meetings. So there were a bunch of folders bunch of directories were not really, nothing really to highlight in them. I've just created them so that people are directed in certain, pushed in certain directions. Again, we have our group papers, and I've updated this week. There's another paper that I've kind of got up here. It's Behavioral Emergence as Explorations of the Possible, and this is something that's pretty wide open right now, but what I'm thinking about is what happens when embryos start to emerge behavior. So what I mean is that the embryo, develops into this larval organism or this juvenile organism, depending on the species. And as it does so, you end up getting these neurons that form. They become, mature enough to start firing action potentials, and they have gap junction connections between them. But then they also need to generate axonal connections and synaptic connections between the cells. And so that happens a little bit later in development. And then eventually, sometimes like in humans, you get an overproduction of these connections. And then in the juvenile phase, you get a pruning of the connections to refine behavior. And so what this is about is it's sort of developmental behavior, which is to say what happens during this process of connecting or booting up the behavior. And so we've talked about that in OpenWorm, where we talk about booting up the worm. And people don't really think about it that way. We think about it, it's largely been thought about in terms of how do you just build a worm that behaves and does stuff. But one of the reasons why we have DevoWorm in the first place is because we want to figure out, how things get there in development. And so, as it turns out, it's not easy. There's a process that goes on, and you get cells that differentiate, then you get connections between the neurons, and you have a body that's developing at the same time. And so in C. elegans, you get this. Well, let's just use C. elegans as an example. You get these neurons that form. They differentiate into neurons, and then they start to form these electrical or synaptic connections, or axonal connections, and then synaptic connections. And then you start to boot up behavior. But even before that, you get things like muscle twitching. and you even get cells moving around. But that's not really behavior, like, goal-directed behavior. But you do get this sort of muscle twitching that's observable, within the egg. And then once the organism hatches, then it starts to behave and crawl around and do all this stuff. So by the earliest larval stage, that behavior is somewhat established. I don't even know really people have studied that deeply, how, like at the very first larval period of C. elegans, L1, what the differences are in behavior between that and the adult. I'm just assuming here, and I don't know, there might be some literature on this even, that it's more or less the same. It's pretty stereotypical, but it's, the idea is that you get these sort of crawling behaviors, these turns get exhibited by the larval worm, very earliest larval worm once they hatch. And then the question is, but how do you get to that point? Even when sort of in the latest stages of being in the egg, how does the worm get to that point? So that's what the paper is going to be about. So we have like the basilaria non-neuronal cognition. So these are the diatoms that we're looking at, trying to figure out how they move. And so that's the group meetings. These are the open papers. So if you want to contribute to that, you may. Then the issues here in this repo. Let's see what we have here. Maybe I can draw some attention to some of these things. Okay, so in progress, we have a bunch of things in progress. Revitalize Friday's Coffee Hour Hackathon. We're doing that now, and it's been somewhat successful. Some weeks we get people, some weeks we don't. This is one of the weeks where we didn't have anyone show up, but that's okay. It's pretty broad. I try to cover issues I don't cover during the week on this, in this meeting when we don't have anyone. Model of motile cells and embryos. So this is actually, I think, connected to the GSoC stuff that we're talking about with Mayuk. We still have the axolotl embryo animations and segmentation. It's kind of been put aside for a while, but I keep bringing it up. I keep hoping to get back to it myself, and I haven't been able to. But I think there might be some interest in it from other people as well. The complexity measures, again, this is where we're looking at creating measures for biological complexity, maybe behavioral complexity, and kind of coming up with things that are robust, like information theoretic measures or other types of metrics that will allow us to gain some insight into these processes. We haven't gotten too far on this. I know George Mikhailovsky presented on this several weeks ago in our group. He had a paper on what he calls complexification. So we have a collection of materials on Google Drive here. I think it's actually, I don't want to share all of it on GitHub, but if you need permission, just click on that link, go to the issue 3, click on that link, and then request permissions if you want to work on it. And then there's some papers here that are kind of in progress. As I showed you, they're open papers. This is the this idea of more error patterns, morphogenesis, and perception. This may be something that will be presented this Monday, so stay tuned on that. I'm not quite sure how I'm going to present it to the group, but I hope to. Krishna Katyal, so these are things on hold. Krishna Katyal said he wanted to review a paper, so I'm still waiting on his slides on that. I think he wants to present this Monday, so stay tuned on that. Create Neuromatch times with DevoWorm, and I'll talk about Neuromatch in a minute. But this actually is something that we can talk about as well. Discuss AI innateness. So that's something that's pretty open-ended, but we were thinking of also tying in some of the a lot of the buzz around AI and what's required to build a good AI model with some of the work we've been doing here. And I think there's a lot of synergy there to be had. I think if you look at a lot of the AI work, I think people are maybe moving into the bio-inspired space, but I also think there's a lot we can take from DevoWorm and apply it to some of these AI concepts, or vice versa. Some of the most famous, or you could say infamous, publicized parts of OpenWorm are based on AI. So, I'm referring to the articles where they talk about booting up a worm's brain and putting it into a robot, which is that's something that is, we can create a model of the worm's brain and then, plug it into a robot, because if you can simulate it on a computer, you can use it to drive a robot. But they make it, when people cover this in the media, they make it sound much more like science fiction-y than it needs to be. So, but I think there's some really rigorous applications of this as well. So this biosystem special issue paper, periodicity in the embryo. I mentioned this in our last lab meeting, and I kind of mentioned it at the end of the meeting. We have I didn't put the link to the repository in here, but we have a repository for this. It was a paper we were going to work on last year, I think, and never got to it. But it's a special issue where they're looking for waves and other types of fluctuations in the embryo. And so this is where we're taking a bunch of different sources of data and we're bringing them together to show kind of these periodicities in cell differentiation and division. We're also going to add some simulated data. And so it's going to be interesting. I'm not quite sure what it's going to exactly look like yet, but we've got some things in. It's a repo on a different team. So I'll have to put that link in here. It's issue 41. So if you're interested, keep your eyes on issue 41. Okay, I think the axolotl montaging goes up with this number 25 axolotl embryo animations. And then, I can go through these rest of these, but I think they're probably not imminent, so I won't talk about them right now. Okay, so this is what I wanted to talk about. Notice I talked about, when I went to the main page of the repo, that we have a lot more followers. And so this is what I've been doing this week. So there's this summer school coming up in July. It's the Neuromatch Academy. And so Neuromatch is it's a group of computational neuroscientists who decided during the pandemic to put together a conference series. And so they put together 2 conferences where they invited people from all over the world to contribute, computational neuroscientists. And it was a huge success. Had over 1,000 people at each conference, had a lot of nice keynotes, worked very well, was very fluid. And I presented with Jesse Parent, also presented some things at this conference. And so it was very, very big success. And so they're putting on the summer school, because a lot of the summer schools are canceled this year due to people not wanting to be in a classroom setting. So they're putting this thing online, and they're running, it's a 3-week intensive set of seminars. So you go over a bunch of different topics. Let's see if I have the link to Neuromatch here. I don't know if I have the actual link to, oh, here it is. Wonder why it goes there. Okay, so what we're doing here is, I've decided, and this grew out of our group, one of my other groups, where we have people who were in what they call the interactive track to this academy, and they wanted to start prepping for the actual academy. And so what I did was I set up this academy discussion group, and this just kind of grew out of a bunch of conversations and some social media interaction, and it became clear that it wasn't going to cut it just to have like a small informal discussion group. So, which is fine with me, but we've created this this website, and this website is sort of the gateway to this whole discussion group. So, Neuromatch pre-course. So, Neuromatch has a pre-course on their GitHub repository, and it's just some files for people or some topics that people want to cover. So, for example, if you want to get ready with Python, here's some information about that. Here's the README. So there's some resources in here. It kind of tells you how to prepare programming, math skills, neuroscience. But there isn't a lot here in terms of like either talking to other people or kind of detailed tutorials or any of that. So what I'm doing here in this discussion group is I'm kind of facilitating this amongst a large group of incoming students to the academy. And so we're drawing from this Neuromatch Academy site, this repo that they have on their site. And they have some basic information there. Kate Bonin, who's a volunteer with the Neuromatch Academy, has also produced some tutorial materials. So we have links to that. Not really sure. This is her fork from the pre-course Neuromatch Academy, but I provided it because you'll have things up here that are not featured on the main site. So this is basically tells you how to do Python tutorials without Colab or with Colab, and so she just kind of gives these tips for people who come in with like basic level of programming experience, kind of like what we have in our group. We have some people who have basic programming experience, and people who are expert programmers or pretty good programmers, but don't know much about biology, and so we have this sort of same problem. Okay, so we have these sources that exist, and then we have our own sort of contribution to this. So if a student wants to take this a step further and engage with some of the topics listed in the Neuromatch Academy syllabus. So this is the syllabus to Neuromatch Academy. This is the outline. Oh, yeah, here it is. Okay, so this is the outline that they have. So they're going to talk about model types, model fitting, machine learning, and then they're going to get, week 2 is going to be like Bayesian statistics, linear systems, like linear models, decision making, optimal control, reinforcement learning. So all those things will be in here. Then week 3 we have real neurons, dynamic networks, network causality, deep learning. So it's basically week 1 is to kind of ease people into models as a concept and what you can do with them. Then week 2 is sort of, Bayesian models, linear models, decision making, optimal control being like feedback models, and then reinforcement learning, which is inspired by operant conditioning and things like that. So that's, and then week 3 will be more about neurons and how they work and some of the ways in which we can network them. And then that leads into deep learning. So, it's a really modeling intensive course. So there is this need to know some neuroscience coming in. But I think a lot of it is like learning how to code, learning, how to use the course will be very like sort of basic level. They don't want to assume too much expertise coming in, but you do need to have some background in some things. You can't just start like with total ignorance of everything. You have to have a little bit of background. You have to know basic stuff, and that's why we're doing this prep this prep program, so that people have a decent handle on some of these concepts. That's the goal. And so, people can look at the syllabus and they can decide what they want to what they need to learn more about. So what I've done here is I've set up a discussion group. I've set up a GitHub repository on our own group's GitHub repository. And the goal is for people to interact and then maybe create study artifacts. And so study artifacts are these things that I describe here. These study artifacts include, but are not limited to, creating a Hello World example in a Colab notebook, collections of neuroscience references, so like a bibliography of different references, or sharing resource lists for specialized math instruction tutorials. So the difference between the collection of references and the sharing of resources is that a resource list, you would have here's some resources on the web. You can learn more about this topic. A collection of references would be, what is it out in the literature? What are some good reviews? What are the kind of topics that people are focusing on? That one is maybe a little bit more advanced, but here's Jesse. Hello, Jesse.
Hey, can you hear me?
Yeah.
I was just stopping by fashionably late.
That's okay. We were talking about Neuromatch Academy. I'm actually recording this and talking to the recording, but that I can keep going, and then I'm almost done with this. So I was talking about the Neuromatch Academy. Can you see my screen here to share?
Yes.
Okay. So I was going over the Neuromatch Academy site, kind of describing these. So why don't I go back over this with you, because we kind of talked about this online, but we didn't get into it too much. So, I was talking about study artifacts, and this is sort of the thing that I had mentioned. I didn't really talk too much about it, but this is sort of the thing people can do. They can create things like a Hello World example in a Colab notebook, create collections of neuroscience references, or share resource lists of specialized math instruction tutorials. So basically, if you have something that you might share with people to prepare them for certain topics, or maybe things they want to know more about, that would be what they could contribute. And then that could be pushed up to this pre-course GitHub repository here, which is the one from the orthogonal lab. And then, you could share that with people. So the idea would be you could push a small tutorial or something if you want to know more about a topic, and then, people would get credit for that. And it would be a good resource. And then you can also create explainer videos, which would be where you would create a short description of something on video, and you could record that yourself, or we could record it, and then that would be a good resource as well. So then, I'm routing people. What's going on now is you can submit your email using this form here. And so this links to a Google form, and you fill it out, and you give your email address and your name and everything, your interests, and then you get invited to this Keybase account. So Keybase is like this Slack type platform that is it's like a competitor to Slack, but I'm using it because it's easy for people to interact on. So you get invited to this Keybase account, and then from there people can interact, people can discuss things. I'm actually posting some tutorials there that I have that I'm putting in there, and so it's a good way to get people to study and kind of direct it in certain ways. And then there's some links on this page So there's some computational neuroscience resources. There's the repo. These are some tutorials that I've just posted. There's some stuff on reinforcement learning and generative adversarial networks from the old DevoWorm ML lecture series that we had last year. We have machine learning and TensorFlow, version control using GitHub, some Markdown. People don't know how to use Markdown, and then some Python for beginners. So these are from various places. And then that's the idea. It's I think so I was looking at the DevoWorm GitHub repository, and I noticed they have a lot more followers than last week. So I think people are coming into the area and seeing things, which is good. But I think that the whole idea of a study group is great. I think it's something that's needed.
Yeah, there's definitely seems to be some demand for that, and I think have you had anything directly communicated with the Neuromatch Academy organizers about that? Because I think there's a general need for it, and I think people are just really happy that there's a key base where they could talk amongst themselves, basically. And I don't know. I just actually myself got into the Neuromatch Academy Slack, so I'm kind of seeing some of the discussion, but I have no previous context. So
Well, I mean, they're very busy putting this all together right now.
Absolutely.
They've been. I was actually working on some video processing last week for the group, and so I got a chance to look at some of the materials, and they're great. They have a lot of good materials. I think people coming in though, I asked some questions on the survey where I asked them to say like, what is your expertise and what do you need to learn? So I have those data for everyone that's answered the survey. It's kind of like a aggregate thing where they kind of give an answer. Just give people. I'm going to give that information to the academy organizers so that they can sort of know that like what kinds of things are people when they sign up for the academy, and it's a small sample relative to the whole academy student body, but you're going to have it gives you a rough idea of what people are coming in wanting and needing. I think they run into the same problem that we do in DevoWorm, where you have people with coding background, people with some neuroscience background, people with both, and you have to sort of adjust to that.
Yeah, it's a really, I don't know what you mentioned previously in the meeting or what the context was for your, anything you've been saying here, but I'll just say generally, it's really interesting because so many things are emerging up alongside of it, and it's very cool to see, and you can see there's such a demand, and there's also a lot of people willing to contribute. Like I've been looking a little bit at their GitHub and the Slack and the different efforts that people are putting together, and it's kind of very first time, very emergent, very spontaneous. But I don't know. I think it's a special, nice thing to see. So I'm happy to see that and help out a little bit with it.
Yeah, thank you for helping with that. That was very good.
That's just posting on Twitter, but I may also. I found my way into. I don't know how to describe it other than literally kind of fumbling away into it, but I might be offering some copy editing to some of the materials.
Yeah.
Academy Slack stuff. So that's cool too.
Yeah, it's good, great. Yeah, yeah, it's all sort of bootstrapped from nothing. So it's like everyone wants to get it. I was looking at the syllabus. It looks good. It is pretty computational. Although I've had some people say they want to know more about neuroscience, and I'm not really sure how to what direction to point them in for the neuroscience.
I saw. Well, there were two things. Actually, I'll ask you about this. One of them is that I just put up in Orthogonal Labs, I did put up the Markdown file. I think you saw that for the whatever roles responsibilities. One of the, I think it was either in there or in a related thing. They listed like a Coursera Computational Neuroscience Coursera course at the University of Washington with Rao. And I feel like, that's I actually had looked at that before, and that's a good computational neuroscience thing for it. And I was going to say, when you were showing the, I guess, I don't know what to call it, the landing page that we have for the lab now, Neuromatch Academy, and you're putting some resources on there. Excuse me, I don't know if I should. I haven't really looked at what's there, I guess, entirely. But I don't know if that's something that is a place to add other resources, or if that should be more like put that in on the GitHub page or whatever. Because I know there's some good resources just for like general stuff. Like there's like a super basic 101 level neuroscience thing is like I think it's somewhat deprecated or at least abandoned. But there was like a Harvard edX really interactive. Previously very interactive, but now somewhat interactive. Really good like intro to neuroscience. I'll try to find that again.
Okay.
I can find it. I think it was. Yeah. Here it is. It's still up. It seems it's something like mcb80x.org. Which is Fundamentals of Neuroscience. Like if you type in Harvard Neuroscience 101, it comes up. But it was based off of edX and Harvard's collaboration with them. I think it's still here. But yeah, that was like a pretty good, like if you really have no background, which is it's kind of where I came from originally. But yeah, I don't know if there should be a place to kind of throw those resources at. There's a lot of them. There's a lot of different places they seem to be propping up. So it's kind of Wild West about that even. But if you have suggestions or like any strategy for that, let me know.
Well, I think what I was thinking was having like on this main page, having some resources listed, and I might put it into a table because it's kind of like a list. But like having it there and then having like the files on Keybase so that people, having them maybe sort of redundant so that if people miss it on the website, they can catch it on the Keybase and then they can get access to it either way. Because I mean, one of the keys I found is like people will like some versions of interaction over others.
That's for sure.
Yeah, some people will like Keybase, some people will like maybe GitHub, some people like the website. I don't think we need to put it. Well, we could put it on the GitHub. Just copy it over from the website so that it's in another place. Just so that if someone prefers the GitHub repo, they can get access to it. So
it looks like there's a extra N on the landing page for Neuromatch Academy.
Right, I'll fix that.
I'm just looking at here. But yeah, I think I'll go through the resources we have listed on the landing page and check them out too.
All right.
Yeah, I don't know. I don't want to barge in whatever you're going to be talking about here.
Oh no, we're almost done. I was just kind of chatting here.
Did anything happen earlier in this meeting?
No, I just did like an overview of things. I'll like if no one shows up, I'll just kind of walk through some stuff that I had, so people can watch it on YouTube later. And but there was nothing really that breaking. It was just kind of recapping things where they stand.
The only immediate updates I have is that I am working on the affordance presentation for tomorrow. I think I will have a little bit about that. I don't think it's going to be totally done yet, but I really appreciate the papers that you sent, because one of them really addresses one of my biggest questions about affordances and really ties in with the iClear presentation, and someone that I really kind of want to work with potentially in grad school, because they're basically dealing with cognitive tools, and to that person, they're looking at how interactions through skilled manipulation or tool affect representations and your cognitive abilities. And one of the papers that you sent was like affordances, a rich landscape of something, whatever.
Yeah.
And it looks at how skill-based affordances are somewhat dependent or related on the skills that you have, not just straight up sensory motor environment or sensory motor interaction, but the skills you have affecting and modifying what affordances you perceive or are available. And I feel like that's like really interesting stuff to me. And tied with other stuff that I'll mention later, like it kind of fits with, a lot of my general research interests that I've been posting about and talking about, and like layers of cognition and how they had toggling and what affects the their development. So that's all I'll say for now. I've had a lot of people continue to reach out about Neuromatch stuff on Twitter, which I'm kind of forwarding to the landing page, and I've seen a bunch of people sign up for the Keybase. I get some updates about that.
Yeah.
And one person reached out, I don't remember his name, but someone actually said they're really interested in our lab. And I wasn't sure if they were kind of saying that just because they thought it was cool, or like we're just, pushing their resume out to everybody or whatever kind of thing. They weren't doing that specifically, but
right.
It can happen. But they do seem like they would actually be a good match. Like they have a lot of interest in. They're like very technically involved in like reinforcement learning and relating it to cognitive stuff. So I don't know if they will or have. I'll check to see who it was, but they mentioned like they wanted to do Google Summer of Code stuff, but I guess they missed that, so I don't know. But yeah, that's cool.
Yeah, you can put them in touch. That would be great.
And then, actually, about DevoWorm stuff, obviously it's sort of like super swamped by everything else that I'm doing and Neuromatch. But I still have some actual projects and ideas for DevoWorm as well. So we're kind of at the end of this time, but all the stuff that you mentioned, including there's the non-neuronal cognition paper that I'm interested in for that, but also I do want to be contributing to some of the things that are related to the current Google Summer of Code projects, or at least like DevoZoo, I think it was.
Yeah.
That's still in play for me. It's just not as immediate as Neuromatch stuff has suddenly become. I spent way more time on Neuromatch Academy stuff this week than I planned, but I'm not disappointed about that either. So I imagine you probably would say the same thing.
Yeah. Well, I mean, I think it's a good. I think I'm gonna kind of wait. Like, I'll be probably busy with the Neuromatch stuff myself for a while. Just in terms of like even with our group, I'd like to. I think it's a good opportunity to kind of address some issues and get some momentum going. Just having our own group kind of going over things, and yeah, it's a good opportunity, I think. And so, but once that's over, we'll probably turn more to some of the other stuff that we have out ongoing. So.
Yeah.
Okay. Well, yeah, it's the top of the hour, so well, thanks for showing up, and see you tomorrow.
Yep, I'll see you tomorrow. Hope everybody is doing all right.
Yeah.
I'll see you tomorrow.
Okay, see you then.